Alrighty. Good morning. I think we're about ready to get started here. So today we have with us Dr. Melissa Debiasse, who is a candidate for the UA faculty position. She comes from us from the Whitney Laboratory of Marine Biosciences from the University of Florida. There she is a postdoc working in Dr. Joseph Ryan's lab, and they're using comparative genomics to look at ctenophore ecology. Prior to that, Melissa did her PhD next door at Louisiana State University. There she worked under Mike Helberg, and they used various methods to look at phenotypic differences across sponges and other marine invertebrates, especially with respect to how they respond to environmental stress. So with that, please join me in welcoming Dr. Debiasse.
Thank you. Can everybody hear me? Yeah? Okay. I turned that one off. Yeah, okay. Thank you all for being here today. I'm really excited to be visiting Dauphin Island Sea Lab. This is my first time here. I am a marine lab person, so this opportunity is really exciting. And I've had a great visit so far, and I'm excited to talk to you about the work that I've been doing over the last few years at University of Florida. I'll start with a little bit of background about who I am as a scientist and my career path. I'll tell you a little bit of background about genomics, which is one of the main areas that I focus on. And then I'll tell two different stories about two different projects that I'm working on. And I'll end by telling you about my vision for my future research program here at Dauphin Island. I'm an evolutionary ecologist, and I'm interested in understanding how biodiversity is generated in marine ecosystems. I got my start working with marine invertebrates, sponges in particular, with Mike Helberg at LSU. I stayed at LSU for a postdoc where I worked with Morgan Kelly, looking at the genomics of salinity tolerance in copepods from the west coast of North America. And I'm currently working with Joe Ryan at the University of Florida Whitney Lab, where I generally study evolutionary genomics in a wide range of invertebrates. I'm also very lucky and fortunate to have been involved in some really exciting collaborations outside the Whitney Lab. I have a collaboration with Brad Davidson at Swarthmore College, and we're looking at tunicate evolutionary development and phylogenomics with his group, undergraduate students. And then I also am working with Adam Reitzel at UNC Charlotte to understand sea urchin genomics and phylogenomics. And the last project that I'm involved in is a really big international collaboration to understand the Cnidarian tree of life. My research program combines a lot of different approaches that are really amenable to marine lab life. I spend time in the field, snorkeling, diving, collecting animals. I bring them back to the sea lab where I perform mesocosm experiments to see how animals might respond to stressors. I spend time in the molecular lab generating data. And then the bulk of what I am doing recently is analyzing data on using high-performance research computing technology. And the main focus, as I mentioned, is genomic data, data from the genome and the transcriptome. So what exactly do I mean by that? In every living organism, if we zoom in, we see cells with nuclei. And if we zoom in on those nuclei, we see chromosomes. And if we keep zooming in, we see DNA. We can see the classical double helix structure of DNA. And then if we zoom in even further, we get to the sequence level where we see the A's, G's, C's, and T's, the nucleotides that make up our genome. So across our genome, there are some regions that code for proteins, right? These are our genes, highlighted here in pink. And if an organism needs to turn on that gene or express that gene, the gene has to be transcribed. So mRNA, a copy of mRNA is made from that DNA template through transcription. And then that mRNA is translated into protein. And then the organism can use the protein for whatever function it needs. And so when I talk today, when I refer to the genome, I mean all of the DNA that's in a cell or an organism. And when I talk about the transcriptome, I mean the portion of that DNA that was actually being expressed. Those genes were turned on. And the question that's been driving my research the last 5 or 6 years, and that will take me into the future, is understanding how organisms respond to their environment. And I think about this question over 2 different time scales. On the short term, I'm interested in how individuals respond to their environment within minutes, hours, and days. These are acclimation responses that happen within the lifespan of a single individual. And at the other end of the time scale, I'm also interested in long-term responses, how populations evolve over generations and potentially over hundreds of millions of years. Oh, I think I missed a slide. So we can study short-term changes using gene expression data, and then we can study these long-term changes using DNA mutations, differences in sequences, gene gain and loss, and regulatory changes. And I'm also interested in the environments to which these organisms are responding. They could be responding to their biotic environment. These could be competitive species interactions or mutualistic symbioses. And they could also be responding to their abiotic environment, things like changes in temperature, salinity, pH, etc. And these abiotic changes, I think, are particularly relevant given the amount of anthropogenic change we're seeing across the globe. And the 2 stories that I'll tell you about today are understanding on the short term how sponges and corals respond to their biotic and abiotic environments. And then at the other end of that time spectrum, I want to try to understand and tell you a story about how ctenophores have adapted to life across deep and shallow seas. And I'll start with the sponges. Some of this information will be, I'm preaching to the choir here, but I wanted to just give an overview. On marine ecosystems, we've got 2 competing processes, accretion and bioerosion. Accretion is the process by which organisms like corals and mollusks create a physical 3D structure through the accretion of calcium carbonate. And then bioerosion is the flip side of that coin, where organisms like sponges and parrotfish break down that physical structure. And the particular species pair, that bioerosion species pair that I'll be focusing on today, is the sponge Cleonavarians, which is this brown, lumpy, encrusting sponge, and Parietes furcata, which is this branching fingertip coral here. Parietes furcata is one of the main reef builders in the Caribbean, and Cleonavarians is one of the main bioeroding sponges that overgrows the coral and breaks it down. When accretion is greater than bioerosion, reefs grow. These could be coral reefs or oyster reefs. They grow, they're healthy, they have a complex physical 3D structure, but changes in our environment are likely to shift the balance of these processes. So, ocean acidification is one of the main drivers that might shift this balance. On this plot, I'll be showing the concentration of carbon dioxide on the left y-axis, time on the x, and pH on the right y-axis. And through the burning of fossil fuels, we've seen an increase in the proportion of carbon dioxide in the atmosphere. And because the ocean absorbs a huge proponent of that carbon dioxide, we also see an increase in the concentration of carbon dioxide in the ocean water, represented by this yellow line. And then because of this, we see a decrease in ocean pH. And this is going to cause problems for a lot of organisms in the oceans, but particularly for organisms that have to use calcium carbonate to grow a skeleton or a shell. The question that we focused on for this project is how will ocean acidification influence or change the species interaction between this sponge and coral? And my collaborators on this project include Amber Stubler and Morgan Kelly. Amber Stubler is a new professor at Occidental College. She and I met when we were graduate students at a field course. She's a field ecologist I'm more of a genomics sort of person, and we have merged our skill set to come up with what I hope I can convince you is a really exciting project. Amber showed that acidification increases sponge bioerosion and reduces coral calcification using changes in buoyant masses and physical changes in the coral. This is a photograph of a coral skeleton grown under ambient pH. This is a photograph of a coral grown under low pH. And these arrows represent the site of attachment of a sponge. And you can see just by looking that under low pH condition, the sponge was able to bioerode much more calcium carbonate than the ambient pH treatment. And what we wanted to ask for this project is, based on these physical changes, what's happening at the physiological level? So how does ocean acidification change the coral's response to bioerosion in a physiological way? And then how might the sponge be changing its strategy for bioerosion when ocean acidification is high? And to do this, we collected data from the transcriptome. Essentially, we looked at genes that were turned on or off, gene expression changes. We performed the research at the Smithsonian Tropical Research Institute in Bocas del Toro, Panama. And we went out into the field, we collected sponge and coral individuals. We broke them into fragments, which was pretty cool because of the way sponges and corals are as organisms. When you fraction them, you get genetically identical replicates that you can follow through time. And so we broke these individuals into fragments, we allowed them to heal, and then we put them into their respective treatments. We had corals, which will be represented by the square and the C. Corals in isolation under low and ambient pH. We had sponges in isolation under low and ambient pH. And then we also had treatments where we joined the two together physically by gently zip tying them together in low and ambient pH. So this is our species interaction treatment. And we took samples, tissue samples at different time points. One was before they were in their interaction, and then 48 hours after we joined them. We were interested in getting that initial stress response. And we had 4 replicates of each one of our treatments. This is a photograph of our experimental setup. These blue barrels held water that was pumped in from the bay behind the station. In the low pH treatments, we acidified the water by bubbling in carbon dioxide gas in these CO2 canisters. And then the water was drained into one of these mesocosm tanks, or mesocosm aquaria, where we had all of our replicates. And to collect the transcriptome data, I'll walk you through my sort of RNA-seq workflow. We would take a particular individual, we would put it into its treatment, it would have a physiological response, it would turn on gene expression, and then we would take a tissue sample, extract that mRNA, convert it into cDNA, because cDNA is much easier to work with, and then we would make copies and then sequence those copies. Then I assembled a transcriptome, and then I took all of those transcript reads and I counted them. I mapped them back to this sort of reference. And here in this hypothetical example, the low pH treatment is represented by pink bars, and the ambient treatment is represented by blue bars. So just to give you an idea of how we can interpret these data, in this particular transcript, it was only turned on in the ambient treatment. This particular transcript was turned on in both treatments, but to a much higher extent in the low pH treatment, and so on. So once we have these counts, I analyze the data in an R package called DESeq2, and I'm going to show you the results now. So the first set of results will be in the form of principal component analysis plots. And these plots show how variation among our treatments is partitioned. You can think about this as where they cluster in transcriptome space, or gene expression space. And when treatments are clustered together like this, that means they're very similar to each other. They have very low variation. But like these orange dots, they're spaced across the plot. That essentially means that there's a lot of variation in that transcriptomic response. To get to our empirical data, The blue circles will represent the ambient treatment. The pink circles will represent the acidification treatment. The open circles represent before, and the closed circles represent 48 hours after. And I mentioned before that all of our replicates are genetically identical. And so we can track the response of an organism through time. This line is not part of the data, but I've put it there so you can see how one specific individual has changed its transcriptomic profile over time. So all of the data here for the sponge first. We can see that there's a large shift in response to being joined to the coral. The open points are all near the top end of the graph, and the closed points, the after, are near the bottom. We can also see that the shift happens in the same direction regardless of whether the sponge was in a low pH or an ambient treatment. So all of the blue moves sort of from north to south, and all of the pink move from north to south. In the coral, we saw a very different pattern. Not much change between the pre and the 48-hour after reaction. So these corals are not having a very large response to the coral. The coral is not having a large response to the sponge. The other thing we can see in these PCA plots is that variation in the coral is to some extent explained by the treatment. So all of the ambient points are at the top end of the plot, and all of the low pH treatments are at the bottom end of the plot. And these PCAs are a great way to visualize the patterns in the data, but we can also use a more quantitative approach by testing for significant differences in gene expression. And this is called differential expression, asking is a gene expressed more in one treatment compared to another. And this also allows us to see if there's overlap between the genes that are differentially expressed. And I'll display these results in Venn diagrams. So for the sponge, the first thing we see is that in response to the coral, there's differential expression of over 7,000 genes. In contrast, in response to the coral when pH is low, there's a differential expression of only 145 genes. So this is an order of magnitude fewer genes being differentially expressed. But the sponge has a pretty large response to low pH. This is an isolation of the coral. And if we look at the number of genes that are shared, we see 2,500 genes are shared. They're differentially expressed in response to the coral under ambient, and they're also differentially expressed in response to just low pH. And what this suggests is that the sponge is using, to some extent, the same set of genes to respond to two different stressors in its habitat. The coral results mirror what we saw in the PCAs. There's a small amount of change, a small amount of differential gene expression. But there are a couple of patterns. In response to the sponge under ambient condition, the coral differentially expresses 35 genes. But under low pH, the coral only differentially expresses 4 genes in response to the sponge. And it has a little bit larger of a response just to pH alone. And unlike the sponge, in the coral, we see a small number of genes, only 5, are differentially expressed in response to the sponge under ambient versus low pH. So here, even though it's sort of a smaller response, the response that is happening looks like it's happening in different sets of genes. So I've talked to you about patterns of expression and numbers of genes, but the really exciting part that everybody always wants to know and that I wanted to know when I was analyzing my data is, what are these genes? What do they do? Can we think about the function of these genes and how that relates to the response, the phenotypic responses of these organisms? And to try to answer this question, I used a functional enrichment analysis, which asks, is there a certain category of gene that occurs more often than you would expect by chance? So I'll talk to you first about enrichment in the sponge. So here we've got the sponge under ambient treatment. And we saw upregulation of genes that were involved in vesicle creation and ion channel formation. And this makes sense based on what we know about the process of bioerosion in this group of sponges. This is an image taken from a publication by Webb et al. 2019, and it shows the specialized etching cells that sponges use in the chemical part of their bioerosion pathway. This is one of those etching cells here, and it's extending a philopodia to the bioerosion site. And these philopodia have vesicles that are filled with acid. So the cell extends this philopodia, the acid-filled vesicle bursts, an acidic solution is released to dissolve the calcium carbonate. And so in order to have this process happening, the cell would need to be, the sponge would need to be creating these vesicles and moving ions into these vesicles. The next category that was enriched were categories associated with ion binding. And this hypothesis is untested and a little more hand-wavy, but it's possible that when a sponge is actively bioeroding, it's breaking down calcium carbonate. Calcium ions are going to be released into the environment, and the sponge may need to bind them and move them away. I did not see any significant enrichment in the sponge when it was interacting with the coral under low pH, and I did not see any enriched categories in the coral, regardless of treatment. And this is probably a result of just low statistical power, given that the number of genes differentially expressed in these treatments was low. If we go back to our big picture question, how will ocean acidification change the sponge-coral species interaction? I found that under ocean acidification, the sponge is able to bioerode more substrate, but with less transcriptomic effort. So it's working smarter, not harder. And I also found that many of the same genes that are differentially expressed in response to the coral are differentially expressed under low pH. So they're using sort of the same set of genes to respond to different factors. In the coral, we found that ocean acidification dampens its response to the sponge, and that the genes that they're using for different stressors are different. And the big picture questions, or the big picture take-home, I guess, from this, is that ocean acidification fundamentally changes the species interaction both at sort of a physical level and at a genomic physiological level. And this shift could have detrimental consequences for calcium carbonate systems like coral reefs or oyster reefs moving into the future. To shift to the other end of the time spectrum, now I'll tell you about ctenophore genomics and how ctenophores have adapted to life in deep and shallow seas. So ctenophores are gelatinous predatory zooplankton. They are found globally. Most species are pelagic, but some are benthic. They're named for the rows of teines or cilia that line their, or that go down their body in rows called comb rows. So you can see them here catching the light. Most species are hermaphroditic, and you can find them, some species exist at the surface, and other species can be found 7,000 meters deep. So this is a deep sea ctenophore. You can see it's like bright red color. There are two ctenophore genomes sequenced currently, one for Nemiopsis ladii and the other for Debiasse. And today I'm going to tell you about our effort to sequence the genome, the third ctenophore genome for Borrelia ovata. My collaborators in this project include people from the Whitney Lab, and also the National Human Genome Research Institute. And I'll walk you through our sequencing workflow. The first step is to go out and collect animals. This is my lab mate, Alex, and she's using a very high-tech piece of equipment here. This is a Tino dipper. It is a Tupperware attached to a broom handle. We collect these animals. We bring them back to the field, take a tissue sample, extract DNA in the lab. We break that DNA up into small pieces, sometimes using sonication. So we send sound waves through the sample to break it up. Depending on the technology we're going to use to do the sequencing, we might make billions of copies of those pieces. We sequence all the copies, and then my favorite part of the process, depending on the day, is putting it all back together, assembling the genome. We collected two different types of sequence data, short read technology from a platform, Illumina, and we also collected long reads from PacBio, Pacific Biosciences is the name of the company. And these two technologies have pros and cons. The short read data are inexpensive to collect. It has a low error rate. But because the pieces that you get are small, it can be hard to assemble through really repetitive regions. So if you have a stretch of DNA, it's just AT, 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 AT. It's very hard to assemble those small pieces through those repetitive regions. And on contrast, PacBio, very long reads, so it's easier to assemble through repeats, but it is expensive. The error rate is pretty terrible, and you can also run into artificial gene duplications. Ideally, in a genome, you want one representative of every gene, but sometimes when you're assembling this PacBio data, it will artificially give you duplicates. I'll walk you through our genome assembly pipeline. This might not be your jam, so you can zone out for a few minutes as I go through the steps. We trimmed adapters from our reads, we error corrected, and then we filtered out the mitochondrial portion of the DNA. We just wanted the nuclear genome. I assembled 3 different genomes in the program Platinus using slightly different parameters. And then I generated artificial mate pairs. You could think of them as artificial reads in a program called MateMaker. And then I scaffolded the best Platinus assembly using a program called MateMaker. I'm sorry, an SSSpace. So essentially trying to take all of the pieces and put them together to have one long contiguous genome. And then the pipeline for the long read data was a little more simple. We just ran every step in a program called Canoe. It did the error correction, the trimming, and it assembled the contigs, or the pieces of DNA, the genome. So to compare our 2 different approaches, I've got this table that I'll walk you through. We've got sequence length, the number of sequences. Sometimes we call the number of sequences scaffolds. And ideally, when you're assembling a genome, you would want to end up with the number of sequences that equals the number of chromosomes. That's the gold standard, a chromosome level assembly. We did not get there. That takes a lot of effort, but so here, the lower the number, the better. We have a general quality score here. The higher the number, the better. We have a BUSCO score. Essentially, BUSCO is a program that you can compare. It's a database or a set of 303 core eukaryotic genes. This is a set of genes that's present in all eukaryotes. And you can compare your genome assembly to this core set, and it counts how many are present. And so the higher this score, the more complete your genome is in terms of capturing different genes, kind of like a metric of quality. And then here, the number of genes in our assembly with a duplicate. Obviously, there are some genes in the genome that biologically have a duplicate, but we don't want to artificially inflate this number. So our short read assembly versus our long read assembly. Our long read assembly is almost twice the size. It has a much lower number of scaffolds than the short read assembly. The quality score of the long read assembly is much better than the quality score of the short read assembly. The BUSCO scores, to me, are fairly equivalent, but the number of genes with a duplicate in the long read assembly is terrible. 70% of our genes have a duplicate, and we know that that's not biologically realistic, and this is probably reflected in double the size here. So neither of these assemblies are great, and to take advantage of the short read pros and the long read pros, we used ultimately a hybrid pipeline where we took the final short read assembly and we scaffolded it with our PacBio reads in the program Lynx. And to compare that hybrid assembly, we got better. Our size, overall size went down. This is much more comparable to what it should be based on what we know about ctenophore genome size. Our quality score, or I'm sorry, the number of scaffolds is a little bit better. It's less than 250,000, but it's still pretty high. Our quality score is better than the low short read assembly. Our BUSCO score is still pretty good, but the really great thing here is that we got our replicate or our duplicate size down to a more manageable level. And so ultimately with this hybrid assembly, we sacrificed contiguity for low duplication scores. And it's a draft genome currently, but it still is sufficient, more than sufficient, to be able to use it as a tool to test a lot of interesting ecological questions in ctenophores. And then I just wanted to give you a snapshot of how it compares to the two previously published ctenophore genomes. It's a little bit on the smaller size. Again, it's much less contiguous. Its quality score is better than Pleurobrachia, and the number of genes that we predict, protein coding region, is within the range of what we would expect. So the particular question, now that we can get to sort of comparative genomic analyses that I wanted to tell you about today, was what's the genomic basis of bioluminescence in ctenophores? Most ctenophores are bioluminescent, including Borrelia ovata and Nemiopsis ladii. And unfortunately, the Nemiopsis ladii video will not play, but it's not that impressive anyway. It's kind of dim. The bioluminescence just goes in like thin little strings down the sides. It's kind of like a dull grayish color. But in contrast, I'll play it again. Borrelia ovata has a really impressive bioluminescence. It goes all across its body. It kind of flashes. You can see it going across the entire body, not just down the comb row. It's this incredible green color. And it's just much more dramatic than it is in Nemiopsis ladii. And typically, bioluminescence is used for things like attracting mates or prey or for avoiding predators. And a lot of ctenophores, particularly in the deep sea, are highly bioluminescent. Yep. Okay. Bioluminescence is accomplished through the use of photoproteins. And these photoproteins are a special class of enzymes that are involved in bioluminescence. And the light production in bioluminescence is proportional to the amount of this photoprotein in the cell. So the more photoprotein you have, the brighter you can be. And we know from the previous genome assembly of Nemiopsis ladii that it has 10 photoprotein genes on 2 scaffolds. And you can see that they are sort of set up in tandem. Each one of these squares represents a photoprotein gene, and they're clustered. And so Schnitzler et al. 2012 hypothesized that this clustering was potentially important for bioluminescent function. And the idea here, the hypothesis, is that if you need to express genes through transcription and translation, it might be less costly if they're all one after each other in the genome. So for the questions for Borrelia ovata, we wanted to ask, given that it's more highly bioluminescent than Nemiopsis ladii, does it have more photoprotein genes, and are they also clustered? And that is what we found. Again, we don't have a highly contiguous genome, but we do see 13 photoprotein genes on one stretch of the genome, and several other areas where there are 2 kind of coupled together. And so what this suggests is that photoprotein genes have expanded in Borrelia ovata compared to Nemiopsis ladii. And these data also support the idea that clustering is important for their functional bioluminescence. The next thing we wanted to look at was what's the evolutionary history of this group of genes. And to do that, we used phylogenetic trees. We have 2 alternative hypotheses. One hypothesis is that the ancestor had one photoprotein cluster, and it was inherited by Borrelia and Nemiopsis. And if this is the case, the tree that we expect to see has photoprotein 1 from Nemiopsis ladii, most closely related to photoprotein 1 from Borrelia ovata on down. The alternative hypothesis is that there was an independent expansion of photoproteins after Borrelia and Nemiopsis diverged from a common ancestor. And if that's the case, we expect all of the Nemiopsis photoproteins to cluster together, and all of the Borrelia ovata photoproteins to group together. This is our empirical tree, a phylogeny of photoprotein genes. And based on this result, we see that there were at least 3 ancestral photoprotein genes in the ancestor of Borrelia and Debiasse. And we also see evidence for multiple independent expansion events in both taxa. If we go back to our big picture, I showed that there's been an independent photoprotein expansion in these 2 ctenophore species, and that gene clustering is likely important for their function. And this work also demonstrates that trait evolution in bioluminescence is driven by the gain of genes, or gene duplication. So for the last 5 or so minutes of my seminar, I'd like to give you my vision for a future research program at the University of Alabama and Dauphin Island Sea Lab. My research moving into the next 5 to 8 years will continue to focus on understanding how organisms respond to their biotic and abiotic environment over short and long time scales. And the first project that I will tell you about is one to predict the fate of bioerosion under global change. I talked mostly about corals and sponges and their biotic interaction, their bioerosion interaction, but these bioeroding sponges are a huge pest to shell fishery. And given the economic importance of these fisheries, it is important that we try to understand how stressors to shellfish in the Gulf of Mexico might change under future conditions. And bioerosion is just, there's not a lot known about it in terms of its evolutionary history as a trait. And so this is something that I will tackle with this project. The first part of the project will be to estimate a phylogeny and infer the trait history of this group across demisponges. And the phylogenies we have for demisponges now are based on a small number of genes. They might be missing key taxa that are important to this question. They might have low support, so we're not really sure what the relationships are. And so I'd like to improve on the phylogenies we have now and trace the trait history. What I can tell from the phylogenies that we have available today are that these bioeroding clades are not each other's closest relatives. So here I'm indicating a bioeroding clade in green here and here. And if we trace the evolutionary history of this trait, it's likely that the ancestral demisponge was not a bioeroder. And what that means is that this trait has evolved independently at least twice across the demisponge tree, which I find incredibly exciting and fascinating. And it sets up a really awesome natural system to test convergent evolution in this trait across the tree. So, oh, yep, here they are, the independent evolution of this trait. So the second part of this project would be to understand the genomic basis of bioerosion. I could look at gene expression differences between these clades and these clades, see if they're using the same set of genes, see if they're using the same set of pathways. Are they changing their strategy under different stressors? Do they change their strategy depending on the substrate that they're bioeroding? And then the last part of this project would be to try to understand the environmental role of bioerosion. And to do this, I envision bringing different clades of these sponges back into the lab, putting them into a mesocosm experiment, and testing to see what their bioerosion response is depending on the abiotic and biotic factors that they're interacting with. I think that this system is competitive for funding through National Science Foundation, DEB Systematics and Biodiversity. And I'd also love to hear your feedback here about the possibility of this work being attractive to agencies like Sea Grant. And then the second project that I'd like to tell you about is predicting the environmental and genomic drivers of phenotype also in sponges. This is Calais Pangaea. It's a genus present in Florida, the Caribbean, and in the Gulf of Mexico. And pictured here are 4 different species that were described based on their morphology back in the 1800s. And what I showed using genomic data is that we should really consider this one species of sponge with many different growth forms. And what that suggests is that one genetic background is capable of producing 3 or 4 different phenotypes. I also found that variation in the spicules in these sponges corresponded not to the growth form that they came out of, but to the geographic region where they were sampled. And this suggests that environment is also playing a role in determining phenotype. So to try to understand this relationship, the first part of this project would be to identify loci that are correlated with these different growth forms using a whole genome, low coverage whole genome approach. I could look for loci that potentially code for some sort of sponge shape. I would test for phenotypic plasticity by doing transplant experiments, reciprocal transplant experiments, where I can take an individual, move it into a new environment, and see how that affects its phenotype, if there's plasticity there for change. And then the last part of this project would be to rear larvae from one of these growth forms or different growth forms in the lab under different environmental conditions to see if there's developmental plasticity both in spicule formation and in morphology or sponge shape. And these sponges are reproductively active from May to October, and a single individual can output up to 200 larvae a day. And I think that this work would be competitive for funding under the NSF Rules of Life program, because Rules of Life has to span 2 different departments, and given the developmental nature of part of the study and the interaction of genotype to phenotype, NSF seems really excited about genotype-phenotype right now, so I think that that would be a great place to seek funding for this project. And with that, I would like to thank you for attention, and I'm happy to take questions. Yes? The question was, how do I explain the low response in the coral to acidification? And one of the questions people often ask me, well, are you sure you sampled enough tissue? And the answer to that question is yes. When we run the Busco test on our coral transcriptomes, we get really high coverage. So I don't think it's an artifact of not getting an adequate sample. It could be that the 48-hour time point was not a great time point. Maybe they have a big response at 12 hours. Maybe they have a big response at 4 days, and we miss that window. So part of the challenge of these experiments is collecting these data are not super inexpensive, and so you have to sort of limit yourself into what time points you collect. And so the other part of your question was that by breaking them up, Yes. So we monitored the pH through these little hobo monitors, and after we fractioned the corals into pieces, we glued them to microscope slides, and we allowed them to heal for 5 or 6 days before we put them into the treatment. So there definitely could be effects of breaking this animal into pieces, but we tried to do what we could to minimize those. But yeah. Yeah. Right, so I looked, so there were like 30 or so genes that were differentially expressed. And so I individually went through them and I said, what categories are these associated with? That's not like a statistical test by any means. But I did think that it was important to do, given I wanted to have some idea of what was happening. And most of those categories under ambient treatment fell into this negative circadian rhythm kind of categories. And that did not jump out at me as anything, no light bulbs went off. The only thing I can think is that, and this is so hand wavy, I don't know, but by putting a piece of sponge on the coral, it's shaded, and maybe that throws off its circadian rhythm because it's no longer sensing like a diel light and dark, I'm not sure. And then under low pH stress, the only category that popped up was just membrane, which is a very broad sort of, in the gene the way you classify these gene categories, there's sort of like a hierarchy, and you get more specific as you move down, and membrane is like at the top. So it wasn't very informative. Yeah, which is a bummer. Yes. Yes. Right. So for ctenophores, no one is sure why they have bioluminescence. But generally speaking, attracting mates, attracting prey, and evading predators is sort of among the ranges that other organisms use bioluminescence for. I know ostracods, deep sea ostracods, have this really incredible behavior where they like puke out this huge like liquid bioluminescence, and it's supposed to be, they like puke this up and then they scoot away, and it's supposed to detract a predator, that the predator will go after that light flash and they can escape. Yeah, it would be great to do some sort of functional test in bioluminescence with ctenophores, but that's, I'm not aware of anyone who's tried that yet. Yes? Yeah.
So you're talking about all this stuff. So is the sponge, is it a ctenophore, and talk about those levels. So Debiasse, is they see some of that in science, we see kind of thing.
Yeah.
So,
oh, shoot.
So who's the basis of the
Yeah. Play from current slide. Okay. So, I will preface this by saying I do not have a horse in this race. I do not care at all if the sponge or the ctenophore is the first animal lineage off the tree. It is a very important question because it polarizes how we think about trait evolution in animals. It happened so long ago, it might never be, we might never have a truly confident answer. What I will say, though, is that I think the evidence now comes down on the side of ctenophores. This is a graph showing papers across the years. Starting in 2008, Dunn et al. Was the first paper to show that ctenophores were the sister group to animals. All of the ctenophore papers are at the top of the slide, and all of the sponge papers are at the bottom. And one thing to note about this sort of is that this question is interesting from like an animal evolution question, but it's also interesting for people who develop methods, phylogenetic methods. And so they eventually started finding out about this problem here, this Shen et al. Paper in particular. People outside of sponges and ctenophores were like, oh my God, this is a really hard question. Can we figure out a way to answer it? And so people who are not associated with either camp have found that ctenophores are the sister group. And a lot of these sponge papers are the same people. Different first author, but it's the same senior author over and over and over and over again. And one of these particular sponge papers, the methods say, they'll say things like, we estimated a tree and we got ctenophores as the sister group, so that was wrong, and we went back into our data matrix and we had a hand-curated set of genes, And now here's our correct tree. Sponges are sister. Which is a little, I don't know, that's not how you're supposed to do science, right? So my kind of gut goes down on the side of ctenophores. The other thing that I appreciate is that we've thought about sponges as the first animal lineage for a really, 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 really long time. Yes, 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 yes. And we also think, maybe not in this room, but sometimes I think we think about animals as being like less complex and more complex. And so sponges, they sit on a reef, they filter water, they don't do much else. And I think that does a disservice to think about them as not being a complex animal. And ctenophores have muscles, they have neurons, and people think there's no way the first animal had those traits because that's too complex. And then if they are the first animal, why did sponges lose those traits? That seems crazy. Why would you lose a nervous system if you evolved it? But the loss of traits happens all the time. And a prime example of that is like in parasitism. If an animal becomes a parasite on another, it starts to lose some of its traits because it can take advantage of those traits in its host. So the short answer is I think ctenophores are the sister group to animals, but the data are, it's a hard question. It's a really hard question. So, yeah. But I still love sponges very, very much. Yeah. Okay. Thank you.